this morning's sessions gave us the overview, and it's really, your work begins now. Okay, so your work begins now at two presentations here that I know many of you have been awaiting, and then we'll have a good panel discussion after the second presentation. We'll have a break in between the two, and then we'll come back and have a panel discussion with 5 folks here, and then that'll set the stage for tomorrow's talks as well. So, Michael Lacey, to you.
So, my name is Michael Lacey. I'm a biologist with the Department of Fish and Game Fisheries Branch in Sacramento. And today I'm going to be talking to you, giving you a little overview of the kinds of things that, of genetic principles and population genetics principles that you can apply to recovery programs. And specifically, we're going to be talking about things that apply, hopefully, to the Shasta River Recovery Program. So in today's talk, I'm going to give you a background about population genetics principles, and then I'm going to address some of the questions that I've been asked to address throughout this talk. And you can see those up there. I won't go through them. I'll call attention to them when I get to those slides. And I'm going to talk about everyone except the one at the very bottom here, the expectations of the time needed. That one is very site-specific, and we can talk about it in the panel if you'd like, but I'm not going to talk about it during the talk, just to let you know, so you're not waiting for it. So genetics is fundamental to recovery programs and to long-term conservation of species because it's a characteristic of healthy populations. Genetic diversity represents the basis for adaptive evolution and is evidence of local adaptation. And also, loss of diversity is a serious threat to long-term species survival. What we're going to be talking about today, at least in part, is population genetics. It's a realm of science in which we look at the behavior of genes in populations, and it's fundamental to the conservation of species. The goal of conservation is to keep the building blocks of evolution to increase the probability of recovery, to preserve ancestral and historical lineages, and amplify the right fish or the right genotypes, and then lastly, to attain long-term viability. All of these things have to be done in order for us to say we've conserved a species. The kind of thing we're trying to avoid is a thing called, is embodied in the concept of the extinction vortex. An extinction vortex is a situation that arises through a combination of genetic traits and environmental traits that function to cause a species to gradually decline to the point of extinction. It works through fragmentation of populations, metapopulation degradation, low effective population size, and loss of diversity. And I'm going to talk about most of those, all of those things as we go along. So there are 2 aspects of genetic diversity that we'd like to call attention to in this kind of a discussion. One is having to do with geographic population structure and the kind of diversity that you see among populations or between populations. And the second is the level of background diversity that you have to work with within a single population. So in order to begin discussion of conservation of genetic diversity, you need to know the relationships among stocks and how much diversity you have to work with within each stock as you target those for conservation. Homing of salmon results in the production of local populations, and those local populations are distinct in that they, individuals within them have a tendency to mate among themselves more than they do with individuals from outside populations or populations that are more distant. A variety of genetic factors then result in those populations becoming distinct from one another, and you get this geographically structured population scenario. Now, generally, distant populations are more different from one another than neighboring populations, and differentiation is often cited as evidence for local adaptation. Because of this differentiation of populations on a geographic scale, local populations are often thought of as being the best for recovery, to focus on for recovery. And in terms of production of hatchery stocks that are based on locally adapted populations are much better, are much more successful than hatchery programs that rely on out-of-basin stocks. That's generally true, and it's something to keep in mind. So you can see that the strategy for retaining genetic diversity or for managing genetic resources depends on this degree of geographic structuring. The loss of a single population in a relatively undifferentiated species might not be very consequential, might not mean much, but the loss of a single population in a highly differentiated species, on the other hand, could represent the loss of a substantial portion of the total genetic diversity. So genetically distinct populations are a characteristic of salmon, and so retaining local stocks is generally thought to be important. It's important. The second aspect of genetic diversity is the amount of diversity that you have to work with within a single population. And this depends on the states, both the historical and present day states of a variety of things. Abundance, whether or not that population has gone through bottlenecks, that is drastic reductions in population size, and whether the pattern of migration and metapopulation dynamics that is being experienced by that population as well. Population size is one of the most important things that determinants of the ability of a population to retain genetic diversity. So small populations are more likely to have high levels of random genetic change, and small populations can have reduced genetic diversity due to the action of a variety of genetic factors, including drift, inbreeding, founder effects, and demographic bottlenecks. There's lots of ways to count the number of individuals in a population. You could just count them. You might be interested in the density of that population. But for genetic purposes, we use a measure called effective population size. Effective population size is used to express information about the rate of the expected rate of random genetic change that occurs in a population, and it's also the size of a hypothetical population that has the same amount of random change due to drift and inbreeding as the actual population experiences. That's not so easy to understand, so I put up an example. Here we have a population of 100 individuals, but it has an effective size of 10, which means that even though that population has a count of 100, it's acting like a population, genetically, like a population that has 10 individuals in it. This population's acting much more like a smaller one, genetically, and the population is also losing genetic diversity at a higher rate than we would expect. There are a number of factors that affect effective population size. These include sex ratio, change in population size over time, and variation in reproductive success. Because these things are always acting in nature, the effective size of a population is generally less, and frequently much less, than the counted number of individuals in that population. So if you go out and you count a population of 100 individuals, you really have an effective size that's likely much lower than that. And that's something to keep in mind, because the effective size is what determines, to a large extent, the diversity that you have remaining. So larger populations have a tendency to retain more genetic variation over time, and very small populations lose diversity much faster than large ones. Here you can see in this graph that up here at these large effective sizes, population size lost over 10 generations is not very much. Whereas in the range here from 10 to 1, population size or genetic diversity loss is pretty great. So one of the principles that you can come up with from all of this is that if you want to retain diversity, you need to start with a larger population. That's great, except that in all the cases when you're starting a conservation program, you typically are starting with a very small number of individuals. The reason you want to start the conservation program in the first place is that you only have a small number of individuals remaining there. Okay. So how many individuals do you need to retain diversity? Well, it's very difficult to tell for any given situation because it's very site-specific, very specific to the recovery scenario that you're looking at. But there is this thing called the 50, 500, 5,000 rule. It's also sometimes called 5,500 rule or the 55,000 rule. And it simply says that the effective size should not be allowed to drop below 50 per generation to prevent the immediate effects of inbreeding, 500 per generation for avoidance of long-term loss of variation due to drift, or 5,000 per generation to maintain adaptive variation. These are not hard and fast rules, so it's instructive to try to see how you might look at this or how we might view this. If you had a population of 50 individuals per generation, we may judge that minimally adequate to avoid the immediate effects of inbreeding. However, we'd be less confident that that population would be able to maintain adaptive potential over a longer period of time. That's the kind of way we use it. Oftentimes, we also use it to set hypothetical targets, and that 50 per generation target is frequently used in that way. So I was asked to talk a little bit about effective size for source populations, and so here's some of that information. Sources for artificial propagation programs should always have as high an effective size as possible. That's nice to say, but like I said before, typically you have very small numbers of individuals to work with when you're starting a conservation program. So it's a very difficult thing to deal with. Still, you should attempt to construct your program so that you collect broodstock in a way, so that you have flexibility in collecting broodstock, so that you have opportunities to increase effective size through that program. You also have the problem that producing large numbers of hatchery individuals from a small number of parents can cause a reduction in the combined hatchery and natural population effective size. And this is called the Ryman-Likely effect, and it's been known to be common for some kinds of hatchery programs. It's to be avoided. Another issue is for conservation programs is the fragmentation of distribution and metapopulation dynamics. A metapopulation is a set of populations connected by some amount of migration between them. Declining species frequently have fragmented distributions, and they have impaired metapopulation dynamics. And what I mean by that is that some species or a population that once had connectivity among a variety of populations then loses that connectivity, becomes isolated, and develops an extinction trajectory that leads it deterministically towards extinction. Here are some possible dispersal patterns and the metapopulations that can result from them. And this is a very hypothetical sort of thing. It's not like you see all of these in salmon or anything, but what happens is if you have intermediate to frequent dispersal in these kinds of cases, you can have the rescue of some populations or the movement of some populations from one place to another. In this one, this is the one you want to avoid, the situation in which populations are isolated and there's no arrows between them, so there's no migrants moving from one to the other, and they can become deterministically extinct. So metapopulation persistence also depends on the balance between colonization rate and extinction rate. Straying in salmonids is known to decrease with distance from the natal stream, and so fragmentation and isolation just decrease the probability that a distant source could provide strays that could repopulate your stream. What this means is that once you've entered a state of population fragmentation or distribution fragmentation, the kinds of situations where you might get the rescue of your population by migrants, the probability of that goes down. Coho salmon have an almost obligate 3-year life cycle, and so natural reestablishment of an extinct brood year can depend entirely on strays from other places in some cases, and that causes more problems for coho. I was also asked to talk a little bit about genetics of source populations and preserving genetic integrity. First question that you'd need to ask is whether there's even anything left to preserve. And much of this has to do with the small population dynamics that we've been talking about. Very small and isolated populations can be functionally extinct even before all of them are gone. That is, they can lose their role in the ecology of the system in which they are found. Very small populations over all brood years may not retain any significant population level diversity because of the time over which they have been small, and because of the fact that there just are a few individuals. It's down to individual diversity at that point. Very small populations may not retain adaptive features. They may have lost that over time, and they may not have the adaptive potential to grow into a population that could persist. And amplification of small numbers of individuals can cause more harm than good, again, having to do with the Riemann-Likre effect. Another question is whether there's potential for natural migration from nearby genetically and ecologically similar stocks. This is, as I said, this is a feature of fragmented distributions, and that's frequently the case in situations where you want to develop a conservation program. Finally, there is potential for natural stock improvement in association with artificial propagation supplementation over time. And I'd like to emphasize here that a phased approach is required in such situations, and maybe all of these situations. It's frequently thought that when you identify a population decline or you identify a conservation problem, that you can do something and fix it. The problem is that you typically can't get from where you are, which is low population size, fragmented distributions, low genetic diversity, and move from that place to the place where you want to be. High genetic diversity, large numbers of individuals, unfragmented distributions, connectivity among populations. You can't get there in one step. And so I would encourage you to envision that there has to be a phased sort of approach to this kind of recovery, where you look at intermediate steps that you think you can really attain, move towards that, assess whether that is really working, and then move ahead, because you're not going to get there in a single step. I was also asked to comment on the balancing of genetic integrity of target stocks while increasing population size and reducing demographic risk. So, with careful genetic stock management, like we heard about for the Iron Gate Hatchery Coho program, you can minimize inbreeding and drift with this kind of genetically informed spawning. And that actually works pretty well. You can also maximize the effective size in the hatchery, and sometimes you can even pair that with the effective size to the number of breeders ratio, keeping that above 1, which is, or at or above 1, which also maintains genetic diversity. Sometimes you can do both of those things. And you can use a phased approach to gradually increase the number of natural origin spawners, the natural origin incorporation in the hatchery, and have realistic natural origin spawning targets. One of the big features of that cause loss of diversity is inbreeding. And of course, inbreeding is mating among close relatives. It's associated with low abundance and low migration. In virtually all cases, inbreeding causes a reduction in fitness. I don't know of any cases in which that isn't true. and this is called inbreeding depression. For small populations, if you know you have small populations, you should probably assume that inbreeding is an issue, or it's going to be an issue at some point. Inbreeding is much more of a problem in small populations than in larger ones. And here you can see a graph that kind of depicts that. So after 50 generations, a population of size 5,000 has an inbreeding coefficient of about 0.5 or so, whereas a population of 20 has an inbreeding coefficient of about 0.7, much higher. The converse of inbreeding is outbreeding. And outbreeding depression is a reduction of fitness that results from mating among unrelated individuals or distantly related individuals. And this occurs due to a loss of local adaptation, a loss of alleles, or a breakup of favorable gene combinations that work together to confer fitness. In most cases, this is related to distance. More distant populations of animals mixed together have a higher chance of causing outbreeding depression, but that isn't always true. Sometimes it can happen with neighboring groups. There's also another issue to deal with is outbreeding, or to keep in mind, is outbreeding enhancement. And this is what you've heard of called hybrid vigor. And in this case, what we're talking about is mixing an inbred stock with a different stock that masks the expression of deleterious recessives in the first generation offspring. Now this is something that may or may not persist past the first generation upon recombination and backcrossing. Hybrid vigor can turn into what's called hybrid breakdown, and you can see the effects of outbreeding depression. And if this happens, it can get worse with each succeeding generation. So it's a little bit insidious. And here we'll talk about, let's go with this. One of the main questions in conservation biology today is whether you should keep genotypes of parental populations intact, or whether you should introduce different genotypes from other populations to deal with the cumulative effects of inbreeding. When you have an inbred population, you don't have a lot of choice. You probably will need to do something at some point, and that means outbreeding. The idea of genetic rescue, the idea of genetic rescue, or this genetic enhancement, is a bit controversial. It's been reported in some species, in Scandinavian wolves and some others, Florida panther, some others. But it's very controversial as to the way that it worked, or whether it even worked at all in some cases. If an impaired population is very small and has been small for a long time, then the existing genotypes may have very limited recovery potential, and you may be forced to move on to an outbreeding scenario, which we have in some cases. I was asked to talk a little bit about the risks of inbreeding and outbreeding. So to summarize here, kind of when a population is small, you should assume that inbreeding is going to be a significant problem. Artificial propagation that eliminates mate choice has a tendency to exacerbate inbreeding potential because you're taking the choices away from those individuals and making them yourselves, and we aren't good without using some kind of a genetic measurement scheme, beforehand to establish unrelated spawners. We aren't very good at mating them, certainly not as good as they are themselves in nature. Inbreeding can be minimized by careful genetic management of spawning. However, and this is important, even if you can minimize inbreeding of your brood stock, if the background diversity is low, then inbreeding is still going to be an issue. And what I mean by that is you can't use a genetic spawning matrix, for example, to get yourself out of a low diversity problem. You'll still have that low diversity background unless you have genes come in from someplace else. To continue this, a Although using a native stock is preferred, if the stock has experienced reductions in population size, if the population size is low, or if the potential for natural migration is low, or maybe all those things are happening, then outbreeding with a nearby genetically similar stock might be considered. You have to consider the potential for loss of diversity through outbreeding depression and weigh that against the cost of inbreeding. And you have to look at the potential for outbreeding enhancement. You have to look at the potential that outbreeding enhancement will be a positive thing and weigh that against inbreeding and outbreeding depression. Further, we have the risks of hatchery supplementation. Hatcheries have the potential to cause fitness and productivity losses in both the hatchery stock and the natural origin stocks that they mix with. And this happens through the mechanisms, some of the mechanisms we've talked about, inbreeding and outbreeding and the elimination of mate choice. And also there's the feature of domestication selection, which is selection in captivity for features that are advantageous in the hatchery, but may not be advantageous in the wild. And also there's relaxation of selection, which is kind of the point of the hatchery in the first place, to cause the early life history or early life stages to have a higher survival than they normally would. Kind of the whole point. Domestication is natural selection that operates during artificial propagation to produce adaptations to the culture environment. It typically requires more than one complete life cycle to produce a permanent response. And domestication selection tends to eliminate fish that can't adapt well to the captive environment, which may include some fish that also are well adapted to their natural environment. So you get kind of losses all over the place. The risks and benefits of using hatchery stocks for recovery kind of go like this. Domestication selection and other mechanisms can reduce the productivity of the natural origin spawners. And you have to consider, oops, turn myself off. And you have to consider whether the natural origin spawners would have a better chance of recovery without any influence of hatchery fish at all, and try to determine whether that would be the case. It is possible to get a demographic increase, plus possibly an outbreeding enhancement genetic rescue effect from adding hatchery origin fish that can compensate for reduced productivity of the natural origin stock if it is small. However, at some point, again, this refers to the phased approach that I've talked about. At some point, you have to transition from a large hatchery origin population and a small natural origin population, and reverse that. Go to a small hatchery origin component and a large natural origin component. Otherwise, you just have a hatchery run, and it could result in the extinction of any non-hatchery influenced natural origin components or spawners. So just to summarize a little bit, source population should ideally be native fish from the stream where supplementation is to occur, and the hatchery population should be integrated. Small target populations are going to pose significant problems for recovery. If it's not possible to use native stock from a stream, then genetically and ecologically similar sources can be contemplated. Imperiled stocks almost always already have experienced this drastic reduction in population size and loss of genetic diversity that goes along with it. And so in many cases, when there are few to no individuals returning each year, it may be debatable whether there's anything left to save. Now, that doesn't mean that you would, if you had a small number of individuals native to a stream, that you'd want to just throw those out and completely replace them with something else. Those individuals could be used as the basis of a stock recovery program, or they could be used as an incorporated element to a stock recovery program. And that's it.
Thank you very much. Please let's thank Michael. Okay, we have time for some questions. I'm sure there are gonna be some.
Hi, Michael. Is there a litmus test for determining if a population is inbred currently, other than the indicator small population size? Other indicators?
It's like I said, you don't. You can do that, but in many cases, you may or may not see the effects of inbreeding right away. But in many cases, you do. You'll see the production of structural abnormalities, developmental abnormalities in offspring. And this is the kind of thing that if you're looking, say, in a hatchery, if you're looking at it, you can frequently see it. You can also measure genetic diversity directly within an individual, and you can get at it that way as well.
Which is how you would look at it before you start a supplementation program. You're looking at depressed natural stock. That's how you would do that genetically. Yeah.
So if you determine, if you did an assessment of diversity in a stock that you wanted to perpetuate, or you wanted to, and you found, for example, like was found for the Russian River Coho program, that the brood stock that we had to work with, the captive brood stock we had to work with, was probably derived from 10 parents, right? Then you know you've got a potential problem. And how big that problem is, well, initially what we thought, because we knew that at the beginning, and initially what we thought was, well, let's wait and see and take a conservative approach to this, and let's wait and see if that results in visible inbreeding depression, or if it becomes a problem. Now I would say something different, though. I would say that you probably should assume that it's going to be a problem, and start incorporating fish from other places right off. I don't think that's the best thing to do, but when you're down to 10 parents, you've probably lost a tremendous amount of adaptive potential, and you're not going to get that back. So I would say that you have to start thinking about outbreeding early. Not to necessarily do it right away, but you have to start thinking about it early.
Well, what would trigger you to start doing something about it?
We found that, well, to answer the question, I'll quote some of the folks who were doing this work, say at Warm Springs. When we started the outbreeding program, the first thing that the hatchery people said was, these fish act just like coho salmon. They act just like they're supposed to. There's better breeding synchrony, there's less abnormalities in offspring, and they generally are easier to even culture. So if you see those kinds of genetic abnormalities, if you know there's low diversity there, I would say you need to be thinking about outbreeding.
Who's next? Michael.
Sunil Madrone with the Matol Salmon Group. In terms of low numbers, we have many watersheds that have that situation with the coho. Many of those watersheds have had stocks brought in from other watersheds in the past. Very few of those watersheds have had that kind of tampering, so to speak, in the past, like the Matol. The Matol is fairly pure strain of coho genetics. Can you talk about the differences between those two situations, and would you approach them any different?
Can you clarify what you mean? What two situations specifically?
One situation is where many of our watersheds, where we have low numbers of fish right now, have already had introduction of other genetic stock from other watersheds in the past. Whereas a place like the Matol has not had that kind of tampering or introduction of genetics from outside the watershed in any significant way. Do you see any difference there in terms of genetics of those two small populations and how you might approach augmentation?
Yeah, there are some big potential differences, but in reality, the similarities of low population size are just things you have to deal with regardless of past history. So, for example, if you had a a native stock that had low population size, or you had a stock that was mixed, but it also had low population size, you could have the same problems in both of them to solve. So from a practical standpoint, right? But there is a difference between a population that is a mixture and one that isn't. A population that's a mixture, you may decide that you don't like that mix, that doesn't fit into the ESU, for example, into the diversity of the ESU, and you may actually want to try to replace that stock in some cases.
Yeah, Rick Costalis with Siskiyou County again. Looking at your 50, 500, 5,000 number there, where 500 looks like a pretty critical number, it would seem to me that the reality of that, which seems to be pretty well acknowledged across the board among everybody, that that's a pretty important number. As stocks trend down or get further from that, below that 500 number, it seems like some kind of buffering strategy on those populations to supplement or augment, whatever you want to call it, those populations, while the underlying problem, whether it be habitat or other issue with that thing that's caught that you're working on trying to rectify that depression, it seems like that would be a real top priority, to be working on some kind of a strategy that would buffer those stocks while those sorts of things can be figured out, given how important that seems to be. I don't get the sense from a lot of these meetings that the department or the agency seems to be really recognizing that and moving as fast as they can to find some kind of solution to buffer those populations in California. These are gone, the Mattole, some of the others are gone. Am I wrong in that perception, that the agencies really are aggressively looking to figure some way to keep things from getting below that 500 number?
So there seem to be a couple of questions there, but one about what the agencies are doing about it. We have addressed it to some extent with Coho salmon, and we are working on it right now. The problem is that the problem with the intervention scenario that you're proposing is that it assumes that it's a foregone conclusion that you can step in, you can increase numbers in some way, that will be easy and cheap, and that it will work. And that's not necessarily the case. There's so many things that are involved in having a program work properly that it's not a foregone conclusion that will work everywhere. The other problem is, so to give you an example of how a conservation hatchery program works. When a conservation hatchery is put in, it takes some time first to say build up that program, right? You may have to build a new facility. You may have to establish a new function for an old facility. You have to get expertise in there at that kind of ability to raise fish. You have to get monitoring involved in there. And all those things mean that you're doing something really new with an existing hatchery, or you're creating another hatchery program. And it typically takes maybe 5 to 10 years to really get good production from those kinds of facilities. At least that's what we've seen here so far in California. So this idea that you could intervene, you can do it quickly, you can jump in there, it's not necessarily that simple to do it right. So I guess what I'm trying to get you to think about is there's a reality to doing these programs that's much more complicated than just stepping in, doing something, and then it's fixed. But the department is working on these issues. The department does not have a lot of resources anymore, and we're working on it as fast as we can.
Well, my point was, if I can just add on,
Yeah.
my point was is that the further you get below that number, the more complex, the more expensive, the more difficult it is. The time to address the problem is higher up on that number scale, where it might be a little more simple. It might be a little bit something, just the ease of buffering, rather than to get it to the program where you're talking about, where hatcheries and other stuff.
To some extent, that's true, and that's why protection of diversity is easier to do than somehow recovering it. Okay, because once it's gone, really, it's pretty much gone.
Dave Hillmer with the Yurok tribe.
Hi, Dave.
So say that you say you've got 2 or 3 small populations that you think have small effective population size, or at least is suspected. Can you combine them to then hopefully increase the effective population size? That's one question. And then my second part to that question, which is a little bit different, is it seems that, well, I'm wondering, I guess, is there some quantitative way to sort of balance the use of perhaps a stock that's gone through a lot of domestication, perhaps originated from a population that's not too similar to adapted to that local area, versus struggling with the small effective population size?
Okay, well, with respect to mixing groups, you can do that. Whether you should or not is another issue. I mean, mixing distinctive stocks is probably not a great idea, but you may be down to having to do that anyway. There are a couple of problems with it. One is that if you mix stocks of different effective population sizes, the mixture, the single population mixture, will have an effective size altogether that's much more similar to the small population than to the larger ones. It's just the way it works. Let me see, there was another issue. Sorry, lost it. What was your, you had another question about?
Well, the second part was in regards to, so in our situation, it seems you've kind of got this choice of, you've got Iron Gate Hatchery, that may have a lot of fish there, they've gone through domestication, they were not from a native stock.
Right.
How do you, is there any quantitative, or is it an art in balancing?
Well, you're stuck, right? I mean, you've got what you've got, and you have to use what you've got. So if you make a decision, for example, to use the domesticated stock, there are a lot of logistical things that become easier because the fish are coming into the hatchery, right? So it becomes cheaper, it becomes easier. That stock can still retain a substantial amount of diversity, and if you can get a relatively large number of individuals from that stock, and you can winnow that stock through natural selection in the stream, then you may be able to recover some of that diversity as adaptive diversity. And but that's very speculative. It's hard to tell. I mean, typically hatchery stocks have lower productivity than natural origin stocks. So when you use one, you have to kind of live with that. Now, if there's nothing there to begin with, it doesn't really matter if the hatchery stock is only 80% as productive as the natural stock, because by adding a number of them, you can maybe make up for that productivity. But the naturalization of those stocks could take many generations. Does that answer the question, sort of?
It helps, yeah. And then,
Okay.
then my other part of it was the balancing it with the small effective population size, but perhaps we'll talk about that more later too.
Well, if you have a hatchery program, you should be shooting for a large effective size in the hatchery, and if you don't have that anywhere, well, then you just kind of have to live with it.
I guess kind of what
You
I was
may.
getting at, there are areas where we have small effective populations that have gone through natural selection, perhaps Scott River, Siam, places like that. And then you got the Iron Gate Hatchery has a ton of fish, but it's gone through all this domestication.
Well, the problem is that selection, if you only have a very small number of fish, then the operation of selection on those fish just reduces their numbers more, right? And if you then have that action of drift and inbreeding occurring over multiple generations, then you end up with not much diversity there to work with. So it's a difficult situation.
I've got a question here, and then I'll take yours, and then we are having a panel discussion. Michael will be involved, and all these questions are fair game. There'll be plenty of follow-up, I'm sure. So Yes.
I'm Craig Tucker with the Karuk tribe. Could you use a panel of SNP markers to quantitatively describe how inbred a population is? And similarly, could you use a panel of SNP markers to guide decisions on the rate at which you add genetic material from a population outside the target population?
With genetic markers of various kinds, you can assess inbreeding, yes. And you can assess the degree to which that inbreeding has been potentially ameliorated by adding new genes from other places. So the answer is yes, you can do that.
So I didn't. So when you were talking about the concept of outbreeding depression,
right?
It seems like using SNP markers as a guide, it would help you make decisions about the rate at which you add new genetic material in a hatchery operation in order to prevent inbreeding. And I don't know if anyone's doing that or that's part of any kind of hatchery genetics or hatchery management plan.
Because the action of outbreeding depression is fairly complicated, it's hard to be predictive about it. But you probably could go back and determine what the reasons were if you really wanted to. You know, what the reasons for your having seen outbreeding depression in a stock that you took action to create. You probably could.
Okay, last question, and then we'll move on to Carlos's presentation.
This is kind of a question and a comment, and I know you're really trying to walk a fine line between having a preconceived outcome of bringing in other fish based on small effective population sizes, and I would be really cautious about assuming that just because you went through a bottleneck or you have a small number of fish, maybe even for a generation, put you in a situation where you don't have a valuable resource left. And I think there's way more demonstrations of populations that have gone through bottlenecks and have been small that have actually, if you relieve the pressures that keep them there, have shown tremendous resilience and rebounded. And so I would be really cautious about just this, looking at the data and saying, oh, we've had 10 fish for 5 years. Because in the Snake Basin, we saw that for lots of Chinook, spring Chinook populations, which are much different than Coho because you have overlapping generations, and the NE is much different and all that. But when we got good ocean and some decent migratory survival, we saw groups of fish that were at 5 and 10 fish go to 300 or 400 in just a generation or two. So just a real caution on what you assume just based on looking at the number. I think you need something else to demonstrate that it's really a problem based on the performance of the fish.
Yeah, I agree with you that's true, and we have examples of situations of both kinds here in California. For example, with winter run Chinook, the population sizes went down to below 200. We had hatchery programs and natural spawning area improvements that caused that population size to increase to as much as 17,000, 18,000 fish. However, you have to recognize that the resultant diversity in those fish is very low. It's still low. And that's something that you can't get away from.
Okay, well, thank you very much, Michael, and we'll move on to Carlos now.
Thank you.